Welcome to the deep dive.
Glad to be here.
Today, we're jumping straight into something pretty revolutionary shaking up biology. AlphaFold3.
That's right. Big news.
You know, we always aim to give you that core understanding fast without getting totally swamped in details.
And that's the plan for today with AlphaFold3, or AF3. It's a major scientific leap.
So for anyone just catching up, what is AF3 fundamentally?
Right. So it's this advanced AI. It predicts the 3D shape of biological molecules.
Okay, like AlphaFold2 did with proteins.
Exactly. But AF3 goes way beyond just proteins. We're talking DNA, RNA, small molecules, ions.
Small molecules like potential drugs.
Precisely. And even modified bits of proteins. Crucially, it also predicts how all these different things interact.
Wow. Okay, so it's not just individual shapes, but the whole molecular dance.
That's a good way to put it. It gives us a much more powerful lens on, the actual machinery of life. A more complete picture.
That sounds like a huge jump from AlphaFold2 then. What's the core change under the hood that lets it handle all that diversity?
Okay, yeah, this is where it gets really interesting. AF2, it sort of worked by predicting the protein backbone first, then figuring out the side chains.
Right.
AF3 uses a totally different engine. It's got this thing called a diffusion module.
Diffusion module.
Yeah.
Sounds technical.
It is a bit, but think of it like this. AF2 was maybe like building with specific Lego bricks.
Okay.
AF3 is more like sculpting with clay or painting dot by dot. It predicts the coordinates for each individual atom directly.
Ah, I see. So atom by atom.
Mm-hmm.
That gives it more freedom.
Exactly. That flexibility is key to modeling all these different kinds of molecules. DNA, RNA, ligands, ions, they all have very different shapes and chemistries compared to just proteins.
That makes sense. A more universal approach. What about the data it learns from? Did that change too?
Sort of. AF2 relied heavily on comparing lots of related protein sequences, these multiple sequence alignments, or MSAs.
Right, using patterns from similar known molecules.
Yeah. AF3 still uses sequence information, but it uses a different module called a pair former instead of AF2's Evo former.
Pair former?
Yep.
Okay. Is it faster? More efficient?
It seems to be. One interesting thing is that it apparently needs less deep MSA information, which could make it computationally more efficient.
So potentially faster predictions, maybe handle bigger complexes.
That's the idea. Faster results, broader analysis capability, huge for research speed.
Okay, so a different engine, maybe more efficient processing. Any other big architectural shifts in how it predicts the 3D structure?
Well, another subtle but important point is that AF3 doesn't seem to need to enforce strict invariance to overall rotation and translation quite as much.
What does that mean in simpler terms?
Basically, it might simplify the AI's internal structure, making it perhaps easier to train on all these varied molecule types without getting confused by the molecule just floating or tumbling around.
Got it. So new core, faster processing, streamlined structure. Let's talk results. What can AF3 actually do better now? What are the key abilities?
The big one is definitely the improved accuracy for interactions, especially protein-ligand interactions.
Ligands, those are the small molecules like drugs.
Exactly. Predicting how they bind to proteins, AF3 is actually outperforming some existing specialized docking software designed just for that task.
Whoa, really? Outperforming the specialists. How do we know that?
There are benchmarks, like one called PoseBusters. It tests how well tools predict the exact pose, the orientation of a ligand when it binds.
Okay.
And AF3 did exceptionally well on this, apparently just using the basic protein sequence and the ligand's chemical structure, often represented as a simple text string called SMILES.
Just the sequence and the SMILES code, not the 3D structure beforehand.
In many cases, yes. It outperformed tools that sometimes even had more structural info to start with.
It's pretty staggering for drug discovery, right? Predicting binding just from basics?
It could dramatically speed things up, yeah. Finding starting points for new medicines.
Okay, so drug binding's a big one. What else?
Protein nucleic acid interactions. So how proteins interact with DNA and RNA.
Which is fundamental for, like, gene control.
Absolutely. Gene regulation, RNA function, AF3 is showing much higher accuracy here, too. Even better than specialized tools like ResetFold2NA.
Another win against a specialist tool. Impressive. Any other tricky areas it's tackling?
Antibody-antigen interactions. That's how our immune system recognizes invaders. Always been a tough nut to crack.
Yeah, antibodies are complex.
AF3 shows considerable improvement over the previous version, AlphaFold-Multimer v.2.3.
improvement, but maybe not perfect yet.
It's still challenging. You often need to generate quite a few predictions and then pick the best one. The accuracy gets better if you generate more possibilities. But yeah, significant progress there.
It really sounds like it's aiming to be a universal biomolecule predictor. Does it cover almost everything now?
Pretty much everything you'd find cataloged in the Protein Data Bank, the PDB.
So proteins, DNA, RNA, ligands, ions.
Yep. And even modified parts of proteins like glycosylation where sugars are attached, or covalent modifications where something's chemically bonded. It provides this unified view.
A unified view of, well, almost everything interacting inside a cell. That's the goal.
That seems to be the direction. It's incredibly powerful.
Yeah, incredibly powerful, huge potential. But let's shift to how it's being shared, because that's caused some discussion, hasn't it? AF2 was very open.
That's right. A key difference this time. With AF2, DeepMind released the full source code, open source. Anyone could download it, run it, modify it.
And with AlphaFold3,
they released pseudocode.
Pseudocode,
meaning a detailed description of the algorithms, how it works step by step, but not the actual runnable computer code itself.
Ah, so you can understand it, but you can't just run their program easily. Why the change?
Well, several factors likely play a role. One is the partnership. AF3 was developed by Google DeepMind and Isomorphic Labs.
Isomorphic Labs, that's the drug discovery company, also under Alphabet, Google's parent.
Exactly. So there's a commercial drug development angle now, which wasn't there in the same way with AF2's academic partnership with MBL-EB.
Right, so commercial interests might mean they want to keep the secret sauce a bit closer.
That's certainly part of the speculation. It changes the dynamics around intellectual property and how it might be used commercially.
So does this affect who can use AF3 and how?
Yes. There's an AF3 server for non-commercial use, free with a Google account.
Okay, so academics can still use it.
Yes, but with restrictions. There are specific limitations, especially around using it directly for drug development pipelines. And there are also daily usage limits on the number of predictions.
Limits and restrictions. I bet the research community had thoughts on that.
Oh, absolutely. There was an open letter published in Nature from researchers expressing, well, disappointment about the lack of fully open source code.
What were their main worries?
The core concerns are about verification, reproducibility, and the ability for the community to build upon the work directly. Open source really fuels that.
Makes sense. Transparency and building on each other's work is kind of fundamental to science. How did DeepMind respond?
They did respond, fairly quickly, actually. DeepMind's research VP stated they are working on making the actual AF3 model, including the learned parameters, the weights available for academic use.
Oh, okay. So not the full code still, but the trained model itself.
Yes, the model weights. They said within about 6 months of the Nature paper's publication, which is a step towards addressing some concerns for academic researchers.
That's interesting. What about Nature, the journal that published the paper? What's their stance on publishing this kind of research without the full open code?
Nature published an editorial alongside it. They basically said their ideal is full openness, methods, data, code for societal benefit. But they acknowledge there can be valid reasons for restrictions sometimes. Lack of data standards, maybe infrastructure issues for huge data sets, confidentiality, safety, legal reasons.
So exceptions are possible.
Yes. Their policies allow for things like releasing detailed methods, pseudocode, or limited code access when a full immediate release isn't feasible. They highlighted the huge role the private sector plays in R&D now.
Right, a lot of cutting-edge stuff happens in companies.
Exactly. And Nature argued it's important to engage with them, bring their work into the peer review process, and publish it for the benefit of the wider community, even if immediate full code release isn't possible.
So they're trying to balance open science ideals with the realities of commercial research.
Seems like it. They emphasized the decision wasn't taken lightly, and they wanted to spark a broader conversation about navigating openness in this evolving landscape, seeking input from everyone.
It's definitely a tricky balance. Okay, so we have this incredibly powerful, albeit slightly less open tool. But no tool is perfect. What are the known limitations of AlphaFold3 we should be aware of?
Right. Important point. It does have limitations. One is stereochemistry.
Stereochemistry, that's like the handedness of molecules.
Left hand
versus right hand.
Precisely. Chirality. AF3 doesn't always perfectly get this right. It can sometimes produce structures with the wrong handedness, or even atoms that slightly overlap, violating basic physics.
So the results need careful checking.
Yes, they use scoring methods to try and rank predictions and penalize these issues, but it's something users need to watch out for.
Okay, what else?
Hallucinations. Because it's a generative model, like some image AIs, it can sometimes create structures that look plausible but aren't real, especially in flexible or disordered regions of molecules.
Making things up, essentially.
In a way, yes. They tried to reduce this by training it, partly using predictions from an older model, AlphaFold-Multimer, which might be less prone to hallucinating in those regions, kind of guiding it.
Interesting. So check the chemistry, watch for made-up bits. Anything else?
Dynamics. AF3 predicts static structures. A snapshot.
Right. But molecules wiggle and move in real life.
Exactly. It doesn't capture that dynamic behavior in solution. And related to that, conformational states.
Meaning the different shapes a molecule might switch between.
Yes. A protein might exist in an open state and a closed state, for example. AF3 might consistently predict only one of those, even if both are biologically relevant. It doesn't necessarily show you all the possibilities.
So it gives a structure, maybe the most likely one it found, but not the whole range of motion or shapes.
Correct. And finally, as we touched on with antibodies, achieving top accuracy for some really challenging targets might still require generating lots and lots of predictions and carefully evaluating them. It's not always a one-shot perfect answer, especially for the hardest problems.
So it's powerful, but needs expert eyes and sometimes significant computational effort for the tough cases.
That's a fair summary. It's a tool, an amazing one, but still needs skilled interpretation.
Okay, let's try and pull this all together then. For everyone listening, what are the absolute key takeaways from this deep dive on AlphaFold3?
I'd say number one, AF3 is a huge leap forward. It predicts structures for a much wider range of biomolecules, proteins, DNA, RNA, ligands, ions, and crucially, their interactions.
A significantly improved accuracy in many cases, especially for things like drug binding.
Absolutely. This holds massive promise for understanding biology better and really speeding up drug discovery.
Takeaway 2. The release model is different this time. Not fully open source, leading to important ongoing discussions about open science, reproducibility, and industry-academia collaboration.
Exactly. That's a conversation that's definitely not over yet.
And third,
while it's incredible, it has limitations. Users need to be aware of potential issues with chemistry, hallucinations, the static nature of predictions, and that top accuracy isn't guaranteed for every target without effort.
So amazing progress, but still room to grow and requires careful use.
Precisely. Continuous development of these tools alongside experiments is really pushing us into a new era for biology and medicine, a more structurally informed era.
Definitely. So maybe a final thought to leave everyone with. As this tech gets better and maybe more accessible, how could it change things beyond just medicine?
That's a great question.
Think about designing new materials, maybe, or engineering biological systems, synthetic biology.
Yeah, material science is a fascinating angle. Understanding and designing molecular interactions, or creating entirely new enzymes or biological machines.
What totally unexpected possibilities might pop up once we can reliably model almost any molecular interaction.
It really opens up the imagination, doesn't it? It's a frontier technology.
It really is. Lots to think about there. Thanks for diving deep with us today.